Chapter 5 reviews the fundamental histology and ultrastructure of nervous tissue. Nervous tissue is dominated certainly by neurons, which are typical eukaryotic cells in that they have a typical range of subcellular organelles, nucleus, nucleolus, endoplasmic reticulum, Golgi, and so on. But the unique feature about neurons is they have long cytoplasmic processes specialized for reception of neural information or transmission of it. The figure that illustrates the neuronal structure is shown on the next page. And you could see the cytoplasm of the neuron containing, again, the typical array of eukaryotic organelles, prominent nucleolus, certainly because these are major cells that produce proteins in the form of neurotransmitters that are destined to be exported from the cell or secreted. Nerve cells are going to have a prominent amount of rough endoplasmic reticulum. And it's so prominent it's known as Nissl substance because of its staining characteristics in Nissl stains, as we'll see. So the term Nissl body or Nissl substance refers to the rough endoplasmic reticulum that is characteristic of all nerve cells. Note, as we'll see, Nissl substance is prominently found, as we show in the diagram, in the cell body of the neuron. It's also found in the proximal parts of a neuronal dendrite, but it is not found in any part of the axon, and therefore neurotransmitter, or the precursors of neurotransmitter produced by Nissl substance and modified by the Golgi, must be transported in the anterograde direction down to the axon terminal. And again, we can see the Nissl substance or Nissl bodies, rough endoplasmic reticulum. A neuron cell body will also have a prominent amount of Golgi apparatus. A distinct cytoskeleton maintaining the structural integrity of the nerve cell is also prominent. This particular neuron happens to be a neuron whose axon is found in the peripheral nervous system because it is surrounded by a wrapping formed by the cytoplasmic extensions of a Schwann cell. And as we know, Schwann cells only form myelin for an individual segment of an individual axon everywhere where the axons are found in the peripheral nervous system. In the next slide, we have a histologic preparation of the neuropil containing neurons and glial cells that are stained with a Nissl stain. Notice we said that Nissl stain will only stain parts of neurons or glial cells that contain rough endoplasmic reticulum. So what we're seeing in this picture is a large multipolar neuron with prominent amounts of basophilic Nissl staining rough endoplasmic reticulum at the arrowhead. Note that in the neuron at the lower right, we see the prominent nucleolus embedded in the central part of a nucleus with lots of euchromatin. Note that letter B illustrates an apical dendrite, again based on the presence of its Nissl staining basophilic cytoplasm. Letter A marks a pale staining area at the proximal portion of an axon, probably at the level of a tapering portion of the cell body of the neuron at the axon hillock. Note that there are a variety of other neuron cell bodies stained in this particular preparation, but the smallest nuclei, there's several of which also shown here, are the nuclei of the central nervous system glial cells, which would consist of astrocytes and oligodendrocytes. At the electron microscopic level, we see an EM of, the cell body of a neuron with a highly indented nucleus, pale staining because of the presence of euchromatin. The arrows probably mark the locations of some of the extensive rough endoplasmic reticulum. And you can see the beginnings of a pair of neuronal processes, probably dendrites, that are branching out on the right and lower part of the figure. At the square over in the corner of the figure, that's an illustration of the surrounding neuropil that again consists of mainly dendrites that certainly are unmyelinated, as well as a structure indicated by the letter M, which is a myelinated central nervous system axon, whose myelin sheath is formed by oligodendrocytes.